Hello everyone. In this lecture, I will be covering the history and structure of DNA. Now, DNA structure was formalized and the paper was published by Watson and Crick in 1958. But almost 9 decades ago, Mendel worked on pea plants and came up with the Mendelian laws of genetics without knowing anything about a molecule called DNA or genes. He explained the laws of genetics or laws of inheritance that are still applicable using the term factors or alleles and not knowing that there is a molecule of inheritance that's present in the nucleus. Now fast forward to 1933. Thomas Morgan, while working with Drosophila, which is fruit flies, demonstrated chromosomes to be an observable unit of inheritance. In his experiment, he was breeding mutant flies, and he realized that some traits can be inherited together, and there are same number of trait groups as there are number of chromosomes. That means group of traits are inherited through each chromosome. Finally, chromosomes, if inherited abnormally, can change, or changes occur in chromosomes can lead to abnormal traits, or what we now call mutations. In 1944, Avery, McCarty, and MacLeod published their research using the word DNA for the first time, and DNA stands for deoxyribonucleic acid. And they published that DNA is a specific molecule that holds the information to transfer traits from parents to offspring during the course of reproduction. Frederick Griffith accidentally stumbled upon a molecule, what he called the transforming factor. And Hershey and Chase confirmed through their experiment that bacteriophages, which are viruses, when they infect bacteria, they indeed inject the DNA or the genetic material into the bacteria rather than the protein shell. And they demonstrated this through their experiment, which I will cover in a couple slides later. Now, let's first look at Griffith's experiment. Now, Griffith was working with a pneumococcus bacteria that contains 2 strains, a virulent or harmful S strain that contained a smooth glycoprotein coat, so smooth for S. And this strain, when injected into mice, killed mice. And the non-virulent R strain, or the rough strain, that does not kill mice, therefore it's harmless. And he also determined that heating destroys the virulence of S strain. So in this critical experiment, Frederick Griffith mixed the heat-killed S bacteria with live R and injected the combination into the mice. And that was his last part of the experiment. But let me first go through the first 3 parts, what he did. In the first part, he took the R strain, which is harmless. So when you inject harmless into the mice is expected to live. And that's exactly what happened. In the second part, he took the S strain, or the smooth strain, which is virulent. When he injected that into the mouse died, as the results were expected. In the third strain, he took the heat-killed S strain. And if you remember, heat when it destroys the virulence of the S strain. So therefore, as expected, the mouse lived. And in the last one is where he mixed his heat-killed S strain with live R and injected the combination into the mice. And the mouse died. Now, this was a surprise because he expected the mouse to live. So when he took the dead mouse's tissue and found that it contained live bacteria with smooth coat or S strain bacteria. So these bacteria were subsequently able to kill other mice and continued to do so after several generations in culture. So he concluded that something in the heat-killed S bacteria transformed the hereditary properties of the R bacteria. The nature of this transforming principle was unknown at the time. Now, Hershey and Chase took Frederick Griffith's experiment, and they wanted to see what this transforming bacteria is, and they performed their extraordinarily important experiment in 1952. And this experiment helped to convince the world that DNA was the genetic material. Alfred Hershey and his assistant, Martha Chase, and they were working at the Cold Spring Harbor Laboratory, showed that DNA is the transforming molecule, not the protein. And they showed it through their experiment in which they took the bacteriophage or the virus and they attached it to the bacterium. And they realized that DNA enters through a tiny hole while the protein coat of the virus stayed out. So this was a confirmation that DNA is the molecule of inheritance that has the capability of transforming other cells and can be transferred from parents to offsprings. In the 1920s, Mueller performed his Nobel Prize-winning research showing that X-rays could induce mutations, and he became instantly famous. But the physicians at the time were not willing to heed his advice, and as a result, they continued the indiscriminate use of X-rays in medicine without any protection. Rosalind Franklin, actually this is Mueller's experiment where he showed how X-rays mutated Drosophila. Rosalind Franklin was a brilliant chemist whose X-ray diffraction studies provided crucial clues to the structure of DNA. Rosalind Franklin and Raymond Gosling obtained this X-ray diffraction pattern, which triggered the idea that DNA was a helix. Many people believe that American biologist James Watson and English physicist Francis Crick discovered DNA in the 1950s. In reality, this is not the case. Rather, DNA was first identified in the late 1860s by Swiss chemist Friedrich Miescher, who identified what he called nuclein. Erwin Chargaff, decades later, researched to reveal that DNA contains 4 nitrogenous bases, and the complementary bases bind as A to T, C to G, adenine to thymine, cytosine to guanine. Now, Watson and Crick were brilliant to use all the prior research and pieced it together to construct a DNA double helix model. Now, in their model, they correctly depicted the 2 strands of DNA to be anti-parallel. That means one runs from 5' to 3' and the other goes 3' to 5' direction. DNA bases are in the middle that are connected by hydrogen bonds, and sugar and phosphate makes the backbone of the DNA. So it alternates as phosphate, sugar, and to the sugar is attached the nitrogenous base, and this makes up one nucleotide. DNA, if you remember, is a polymer. So the monomer of DNA is a nucleotide that comprises of deoxyribose or 5-carbon sugar, a phosphate group, and one of the 4 nitrogenous bases. As Erwin Chargaff revealed that the 4 bases are A to T, C to G base pairing arrangement. So, to recap everything, there are 2 types of base pairs, purines and pyrimidines. Adenine and guanine are the sub bases of purine category, and thymine and cytosine are the pyrimidines. I've also given you 2 links for the YouTube videos to visualize the structure of DNA if you need further understanding. So, once again, DNA consists of 2 strands of nucleotides held together at bases by hydrogen bonds. 2 kinds of base pairs form A to T and C to G. Base with double ring structure are the purines that always binds with base with single ring structure, and those are the pyrimidines. The covalent bonds that holds the phosphate and sugar together are called phosphodiester bond. So this wraps up our lecture on history and structure of DNA. In the next lecture, I will be talking about DNA replication. Thank you.